Hi, everybody. I'd like to talk to you today about a concept called plasmid mapping. Because we're going to start dealing with DNA as we did a gel last week. We're going to try and do another gel this week. But because we're going to be spending so much time doing the gel and the restriction in class, I think it's going to be a good idea to try and do a little bit of outside the classroom learning. So that's sort of the idea behind the next couple of videos that I'm going to try and make for you. This one is going to really talk about a thing called plasmid mapping. And to even talk about plasmid mapping, we have to sort of talk about what plasmids are, and talk about what plasmids are. We got to talk about DNA and a whole bunch of things that let's just get right into. So plasmid mapping is a concept where we're going to sort of construct a map of a piece of DNA called a plasmid. And to talk about that, let's look at a bacteria. So this is a bacteria. This is a cell membrane, and I kind of made it a little bit of a bacillus bacteria, but it could be any type of bacteria. And a bacteria typically has two types of DNA in it. And we know DNA from freshman year is deoxyribonucleic acid. It's double-stranded. It has a double helical shape. It's made of four bases, A, T, G, and C. And it's basically the blueprint for every piece of, it's the blueprint of the genetic information that every organism has. So, it has the instructions basically to build you and me and every other living thing on planet Earth. So what we've done in the past 20 or so years is we've been able to get really good at manipulating it. And that's sort of where this is all coming from. So a bacteria has usually two pieces of DNA. One of them is a mess, but it's a big mess. And just go with the fact that it is, it's a big red line, but it is double stranded and it's tangled. And this is what we call chromosomal DNA, or the bacteria's self DNA. So this thing is self DNA. Okay? And this is what keeps the bacteria functioning. This is what the instructions are for all the enzymes that the bacteria uses to break down food, bring in food, expel waste. If it's a mobile bacteria, it's all the blueprints for the machinery that helps it be moving. This is sort of what keeps the bacteria running. But then, elsewhere in the bacteria are possibly these things, and usually if you take AP biology, they actually tell you they're in all bacteria. I've always read that they are in an overwhelming majority of bacteria, but there are a few bacteria that don't have these things. But they're really small pieces of DNA, a few thousand bases in size, but they're all circular, and they're actually called plasmids. So we've got two types of DNA in a bacteria. We've got the bacteria's self DNA, or the chromosomal DNA, and then we have these things called plasmids. And plasmids are typically pieces of DNA that the bacteria has taken up from its surroundings, or otherwise somehow been introduced into the bacteria. And they might have characteristics or traits encoded on them that will help the bacteria out. And the big one that comes to mind for me is ampicillin resistance. Bacteria, as we know, aren't supposed to be resistant to ampicillin or any of the cillins. The cillins are supposed to inhibit the bacteria's ability to perform mitosis, not really mitosis, but binary fission. And basically, over the years, they have picking up these chunks of DNA from outside that are helping them degrade the cillins so that now they're immune to the cillins, and now they don't work when you get a strep infection or a staph infection or whatever if you're dealing with MRSA. So the idea here is that we've got two types of DNA in a bacteria. Now, how does the bacteria tell the difference between the two? And the easy answer is, for starters, the bacteria, whenever it performs binary fission, whenever it divides itself, there's an enzyme, and don't worry about what the enzyme is, but there's an enzyme that comes on when the bacteria is copying its own DNA, and every little while, I'm making these little black lines. But imagine that these black lines are everywhere on this DNA. These black lines are they're a methyl, which is organic chemistry, but a methyl group. And for all that you need to know, a methyl group is an organic chemical and it's plugged onto the DNA. And that allows the bacteria to realize that this is its own DNA and any other DNA inside of it is not its own DNA. And this is really important when something kind of creepy, such as a phage, lands on the DNA and tries to put its DNA into the bacterial DNA. That DNA doesn't have these methyl groups on it. It's not methylated. That's what the actual term is called, methylated DNA. So this DNA doesn't have any methyl groups on it. And what happens is the bacteria, over evolutionary time, has come up with a way to break down that foreign DNA, and I'm basically making Pac-Man here, so just bear with me. These are things called restriction enzymes. And what they'll do is they will realize that, uh-oh, we have unmethylated DNA in here, and we're going to latch on to that unmethylated DNA. Let me see if I can get these to latch on. Or, yeah, unfortunately. So this guy's going to latch on and pretend this guy latches on, and they're going to run up and down that foreign DNA, and they're going to search for very specific genetic sequences, groups of A's, G's, T's, and C's, and cut them. And as we discussed on Friday, once this DNA gets cut, essentially it's useless. So the phage, the virus that tried to infect the bacteria and take over the bacteria, now that DNA that it put in there isn't going to work because it's been cut. And the big interesting evolutionary part is that this originated millions of years ago, billions of years ago, and we discovered them in, honestly, the late 1970s, and we actually call these things restriction enzymes. Sorry for my bad handwriting, but those little Pac-Man dudes are called restriction enzymes, and what they do is they search for these sites that they can react with, and when they find one of these sites, they'll actually cut the DNA. Now you might be asking, well, what happens with these plasmids? Aren't they outside pieces of DNA? And the answer is yeah, but the bacteria sort of has ways of recognizing these things without degrading them with the restriction enzymes as well. So the idea here is that plasmids are usually good, self-DNA is always good, and phage DNA, viral DNA, is bad. And the bacteria has figured out ways over evolution to break down this bad DNA. So that's sort of the role of restriction enzymes. It was, like I said on Friday, the first immune system for bacteria, if you will. And it's sort of how you guys work also. You know, you have seen, so the bacteria over evolution has seen different bad things trying to attack it, and it has developed very specific defense mechanisms against those bad things. You guys have the same thing. You know, you had chicken pox, you know how defenses against chicken pox, so you shouldn't catch chicken pox again. Or if you got immunized against chicken pox, you can recognize chicken pox and attack it before it really does anything bad to your body. But the way this works is these enzymes will actually make two different kinds of cuts on that DNA. And we should talk about this concept called a blunt versus a sticky end when the bacteria cuts. So, you know, as I said before, DNA is double helical, it's two-stranded, and I'm just looking at a couple of places where let's say some restriction enzyme can cut. And if we're talking about a blunt cut, that would mean Pac-Man, and let's make Pac-Man. Let's go with this. Here's Pac-Man. So this is the restriction enzyme. A blunt cut would mean that the enzyme is going to come in and cut, and just for argument's sake, let's say it cuts here, right here. And you will end up with something that kind of looks like two ends of two boxes, and that's actually why we call them blunt, because they're not sharp. Two ends of two boxes from the DNA, and this is a very clean cut, and that's sort of one way that bacteria restriction enzymes will cut DNA. If you notice, though, the places that these things cut are typically what we call palindromic sequences, meaning they're read the same frontwards to backwards. and I tried really hard to make them, but I don't know if I really did a good job of it. A-T-T-A, yeah, I did it backwards. They're supposed to be palindromes, but they're not, and I'm sorry. But these restriction sites are palindromic sequences. So I guess if I wanted this to be a true palindromic sequence, I can't even fix it. Yeah, I can't. I'm sorry, and I don't want to redo the video. So when you do the reading, you'll see some examples of palindromic sequences. Anyway, so the restriction enzyme will recognize a palindromic sequence, which is something that can be read the same along the top going one way and along the bottom going the other way. I mean, you can probably see what I was trying to do here. Maybe if we get rid of... this will work actually. So the palindromic sequence here is right here. This is a palindromic sequence. So AATAA can be read the same going both ways. All right. And there are some better examples in the book because they're supposed to be read going forwards and backwards on the bottom strand and the top strand of DNA. Anyway, so a sticky end is what happens when the restriction enzyme will cut in such a way that you will end up with these overhangs. So now you'll actually end up with a piece of DNA after this cut happens. So let's say the restriction enzyme comes in and it runs along the DNA and it snips here, and then it breaks these bonds, and then it snips here. So now we're going to have two ends of the DNA, and just deal with my horrible handwriting. From one side of the DNA, you're going to have an A and a T, A, T, A. And on the other side, you're going to have a T, T, A, T, T, A, T. And now these overhangs are on two different pieces of DNA. And the reason this is important is because DNA hates being alone. DNA does not like being single-stranded. It always wants to be double-stranded. And what we can do is, if we get another DNA restriction enzyme to come in and cut more DNA, it will cut at the same sequence, and we can plug into these overhanging fragments new DNA. And that's sort of the idea behind genetic engineering, or at least it was until about until 2018 when we started doing CRISPR-Cas9. But for right now, we are able to use these enzymes to cut other things out of other organisms. And because these are free hanging pieces of DNA, if we can plug something else in that has the complementary DNA base pairing, T, and then we can, so this is a cut end and this is a cut end, and then we have some gene for something important in the middle. We can actually put in a new piece of DNA and basically stitch up these ends, and now you have this new thing inside of this old plasmid DNA. And that's actually really important. This is what we did. So imagine this black box is the insulin gene. So now we've taken a plasmid and plugged into that bacteria plasmid the gene, the DNA that codes for human insulin. And that's actually how we are able to make a ton of insulin, whereas 100 years ago we had to destroy tons and tons of pancreases to get insulin. So that's one piece of the puzzle. Another piece of the puzzle is what we call a single versus a double digest. And now all that really means is a single means you are hitting a piece of DNA with one restriction enzyme. And a double digest or a triple digest would be more than one restriction enzyme. So you're hitting the same piece of DNA with two or more restriction enzymes. Now, why is that important? Well, you can actually get, instead of just one of these sequences where the cut happens, you can get multiple places where these cuts can happen. And that's important because sometimes when we're doing genetic engineering, you need multiple sequences that you need to be able to cut. Again, there's more about that in the book, but this is just sort of an overview of what's going on. All right, so I hope at least this made sense even with my bad palindromic sequences, but realize that palindromic sequences need to be able to be read forwards and backwards. And top to bottom and bottom to top, as well as what the difference between a blunt and a sticky end is. Okay, and also we need to remember how the DNA is differentiated in a bacterial cell. So we have self DNA, we have plasmid DNA, and then we have viral DNA, which is what this origin for the restriction enzymes is. All right, so now let's talk about what we've done in class. And on Friday, we made a restriction, or I'm sorry, an agarose gel. And we ran what we call what the restrictions were. We basically cut that DNA up with different restriction enzymes. And we ran them on a gel. And I think I need to explain to you what actually happens in the gel so we can start understanding what's coming next. So we run a gel, and for starters, we always throw the ladder, which I'm going to call the MW, the molecular weight marker. The ladder goes on one side. And you've got a ruler here because, believe it or not, distance is actually very, very important in terms of understanding what's going on in a gel. So what happens is in every one of those tubes, there were billions and billions and billions and billions of pieces of DNA. And each of those pieces of DNA had a different length to it. And we measured the length of DNA in a unit, this is a weird unit, in base pairs. So we want to know how many base pairs that piece of DNA might be or is. And as you so up here are all of the wells that we loaded the DNA. And what happens is you might have a whole bunch of different molecular weight size pieces of DNA, different base pair sizes. And what the agarose does is it's basically a whole bunch of small holes, we call them pores, and the DNA is trying to get from the negative side towards the positive side. Remember, running towards red. So DNA is actually negatively charged. If you know anything from physics or from 8th grade science, like charges will repel and opposite charges will attract. So the negative DNA wants to get away from the negative part of the gel, which means it runs down towards the positive side. Now, because these are all full of holes, this agarose is full of holes, the pieces of DNA that are small are going to be able to get through those holes faster. We actually call it reptation because it's almost like a snake slithering through these holes. And these big honking pieces of DNA can't do that as quick. So what happens is you get small pieces of DNA that run further and big pieces of DNA that don't get to run as far. And part of the puzzle for figuring out how big these pieces of DNA actually are is we need to know how far they've run. So very, very basically, I'm just going to make different bands that go different distances, and I'm going to make all of this up, but I hope it'll illustrate my point. So we have 10 fragments of DNA. Each of these has hundreds of thousands, if not millions of fragments of DNA in them, but we had 10 of those, so there was a lot of DNA in the original well. And the stuff that doesn't go very far is big. The stuff that goes really far is small. And then we ran all of those different restrictions, and we ended up hopefully with different banding patterns as we went. So, and as we said the other day, if you have two banding patterns that are identical, then you're a DNA match, which means you either did the crime, or you are the father, or your remains have been identified somehow, some way. We can use these pieces of DNA, these fragments, and the size of the fragments, the lengths of the fragments, to do some science, okay, for lack of a better term. So, where does that leave us? We run a gel, and we have on one side a molecular weight marker, and then we can actually identify how big these fragments that are of unknown lengths are. And this is actually what we're going to be doing Thursday and Friday of this week. For today, and Tuesday and Wednesday and Tuesday and Wednesday at least, we're going to be able to tell you how big the DNA fragments are, and we'll use that information. Later on in the week, you're going to actually be figuring out how big these DNA fragments are. So, for example, the molecular weight markers. This might be 10,000 bases. This might be 8,000 bases, 6,000, bases, 5,000 bases, 4,000 bases, 3,000 bases, 2,000 bases, 1,000 bases, 800 bases, 500 bases. So all of these lines, these bands, have a corresponding number of base pairs. Okay? And we can actually tell that by looking at the package insert. Remember how I bought the 1 kilobase molecular weight marker? Well, New England Biolabs, when they manufactured that, they actually basically, when they did a chemical reaction to make those, they know exactly how big they are. So what we can do is we can, this line lines up with 5,000 bases, so we know that this band is 5,000 bases. This line matches up with 2,000 bases, so we know it's 2,000 bases. This one is a little bit more than 8,000, but it's way less than 10,000, so this might be 88,500 bases. This guy lines up with 10,000, so that is 10,000 bases. This one lines up with 5,000. So I hope you're kind of getting the idea that we can sort of do a quick and dirty look at a gel and identify how big the base sequences are. So that's sort of what the gel does. And all we're doing is looking at how big the pieces are of a piece of DNA that got cut by restriction enzymes. And that's the only real reason you use a gel. Now, pieces of DNA that we're typically dealing with are these circular pieces of the DNA called plasmids. And, depending on the restriction enzyme, you can get a cut here, you can get a cut here, you can get a cut here, and you'll end up with a big piece of DNA, a medium piece of DNA, and a small piece of DNA. And you can sort of predict where they're going to land on a gel, or you can use a gel and actually infer how big those pieces are going to be, or how big they are and how much distance is between the cut sequences. So each of these lines is a place on this circular piece of DNA that got cut by the restriction enzyme. And that is what we call restriction mapping. So here we have some real DNA, well, a real gel, quote unquote. And we've got, just like our gels, 1, 2, 3, 4, 5, 6, 7, 8 lanes. And we've actually got sort of a ruler here, millimeters, and you can almost pretend this is the molecular weight marker lane. So we know 50 base pairs goes this far, 30 base pairs goes this far, 20 goes this far, 15 goes this far, 10 goes this far, and 5 goes this far. So now we've done a restriction with Hindi 3 or Hindi 3 and EcoR1. So these are two restriction enzymes, and these were both single digest, which means the piece of DNA, the plasmid, was hit with Hindi 3 and EcoR1 in two separate test tubes. Then we did a double digest, which means we put the DNA in the same test tube with both Hindi 3 and EcoR1. And what we're going to do is we're going to figure out how much distance is between each of those restriction sites. So best way to do it is sort of make a table and sort of look at your gel. I know Hindi 3 in this DNA hit and cut once because I only have one band on the entire gel. So Hindi 3 cut once, and that made this piece of DNA open up. The circle became a line basically. But I know that because it only ran to where 50 base pairs is, that the whole piece of DNA is only 50 base pairs big. Okay? So now I can look at what EcoR1 did. And also, by the way, I've got my little chart here, and all I did was Hindi, EcoR1, and the double digest. Hindi made one band that's 50 bases. Okay? So that means there was only one cut, and that means the whole piece of DNA is 50 bases. There, 50 base pairs. EcoR1, which is another enzyme, cut this piece of DNA into 1, 2, 3 pieces of DNA. So now I know that EcoR1 cuts a circle into 3 bases, or into 3 fragments. Okay? Which means, and I'm going to move my Hindi 3 because I think this is, well, you'll see. So if I want to cut this circular piece of DNA into 3 fragments, I'm going to cut once with eco and again with eco. So now I have 2 pieces and another time with eco. And now I have 3 pieces of DNA. I hope that makes sense. 1, 2, 3. 3 cut sites, 3 pieces of DNA. Okay? Now, the interesting part here is what we're doing when it comes to plasmid mapping is we want to see how much real estate there is between this eco and this eco, this eco and this eco, this eco and this eco, and we want to see where the Hindi 3 fits in. Okay? Does it fit here? Does it fit here? Does it fit over here? So that's sort of the entire purpose behind looking at restriction mapping. Okay? And the way we do it is first, I mean, and this is sort of where I said it's a Sudoku. It's a piece of logic puzzle. So I know eco will give me a piece of DNA that's 30 bases big, another piece of DNA that's 15 bases big, and another piece of DNA that's 5 bases big. So this isn't even to scale. Let's say this is actually up here, and I'm going to turn it. Eco. So now this is a little bit more representative. From here to here is 5 bases. Does that sort of make sense? 15 bases. Again, let's try and make it to scale. We'll move this guy over here. So from this cut to this cut is 15 bases. 15 base pairs. Okay, so now I've got a 5 base pairs sequence here. I got 15 base pairs here. And that means from here to here must be how big? I hope you said 30 because that's this particular band, which is this down here. So from this eco cut site to this eco cut site is 30 bases. From this eco cut site to this eco cut site is 15. And from this eco cut site to this eco R1 cut site is 5. Badly written, but 5. So that part's not hard. The hard part comes with where does this Hindi fit in? Does it fit inside of the 5? Does it fit inside of the 15 somewhere? Or does it fit inside of the 30? And what you can do is you can look at what the double digest is because you now know that I ended up with 4 pieces of DNA when I put the Hindi 3 in there too. I have a 10 base sequence. I have a 20 base sequence. I have a 15 base sequence, and I have a 10 base sequence, and I have a 5 base sequence. So whenever I do these, I sort of make this chart, and then I ask myself, well, what pieces are new, what pieces are different, and what pieces are the same? Because the pieces that are the same haven't gotten cut. The pieces that are new are what happens when we have taken a piece that was big and we've made it smaller. So I know from my Hindi, I lost my 50 base pair. And from my eco, I lost my 30 base pair. I now have a 20, a 15, and a 10, and a 5 for the double digest. So the 2 5s haven't changed, so I know that this Hindi does not go in here. So I'm just gonna make this red and say nothing's going, the Hindi 3 isn't going in there. So my 2 5s were the same. My 2 15s are the same. So I'm not gonna put the Hindi 3 in here either because this piece is identical from the eco digest and the double digest. So what's different? I went from a 30 to a 20 and a 10. So somewhere in here is where my Hindi cut. So what I'm going to do is I'm going to move this over here. And the cool part is you can only be a certain amount right. But let's put this here. Okay. So I know from one eco cut to my Hindi, from eco to eco is 30, but from eco to Hindi is going to be 10 bases. And from Hindi back to eco, that's going to be the other 20 bases. Both of these add up to 30. So I'm still going eco to eco, 30. But eco to Hindi, 10. And Hindi back to eco is 20. Now you may say, but Mr. D, I don't know if it's 20 on the top or 20 on the bottom. And it really doesn't matter. we can only get so exact with this. But what I can do is I can know, okay, we got a 30 base sequence of eco that's been cut into a 10 and a 20, and that is corroborated by the gel. Okay? So that is the first restriction map that I'm showing you how to do. It's very simple. I'm actually going to put another couple of videos in the link from some other people to help you if you want to know a little bit more about this. But we're going to start doing some problems like this hopefully this week, looking at some more complicated restriction maps. But I figure this is a good way to just introduce the material, and without spending too much more class time on the introductory material, I can just start throwing some practice problems at you, and we'll go from there. Alrighty? I hope this helped. I hope it wasn't too confusing. And I would love for you to start reading, like I said to some of the classes on Friday. I'd love for you to start reading chapter 4, because that will really help galvanize this in terms of what's going on. All right, I'll see you in class.